I just put it there.
So now we are starting the next session. So let us all welcome Dr. Graham Ball.
So what I'm wanting to do here is show you some sources of data, some ways of analyzing data, and then ways of getting some interpretation from data. And I think I did a little bit of this the other day, but what I want to show you is, if I just go to Google Chrome, and you can follow this if you wish, is if I Google GONCBI, and if I click on that, this takes me to the NCBI Gene Expression Omnibus repository. Now, in this case, what I'm going to do is I'm going to look for colorectal cancer. But you could look at a whole range of things in here. And there's quite a big repository of data. So if I search for that, you can see there were 36,797 experiments for colorectal cancer. That means essentially 36,797 replicates. So if I click on that, I now get, and I'll zoom in on this because it's not, I now get lots of records. Each one of these records is a data set. And we can look at this in terms of, but in many ways. So first of all, we've got the title, which is good, tells us what somebody has done. We've got the data set abstract. We've got the species. We've got the fact it's expression profiling by array. We've got the platform they use to profile. So this is GPL 15048, which is a Rosetta Merck human RSDA custom Affymetrix chip. We've got the number of samples, which is important. We've got the data types. So we've got the cell files, which is the raw data from the Affymetrix. We've got the CSV files, and we've got text files. So that's useful because the text files will contain the data we need. So I can also do things like filter this. So for example, I want to filter for human. So if we look up here, you can see we can click on that and select human data. Oops, sorry. So now, you see in our search details, it's put Homo sapiens in there. But we could look for microbial data, we could look for animal model data, we could look for cell line data as well. I've clicked on here to look at arrays, but we've got methylation studies. In other systems, we have proteomic data, transcriptomic data, RNA-seq data, DNA sequencing data. So this is now, what I've done here now is I can also rank these. So I can sort by the default order. That's just the age, in a sense, of the study. So this one is a P53 study. But also, if I want to do a reasonable experiment, I want to sort by the number of samples, high to low. So the more numbers, the greater the number of the samples, the greater the richness of that data. So you can see here, some of these are quite good. There are more and more, there are meta-studies where people are looking at multiple data sets in one record. And we use that a lot because what we're doing is comparing each one of those studies separately to find the common features and using them to effectively cross-validate against one another. So, I'm just going to get my notes. So I've looked previously, and I'm looking for an array which has got the GPL 570 array. So that's an Affymetrix HU133 plus 2 array, which is quite one of the most common ones. So you can see here there's a GPL 570 there. So this one has got multiple platforms, and this one is looking at prognostic signature to predict metastasis in colorectal cancer. So we could analyze this data if we wanted to start exploring the processes associated with risk and the like in colorectal cancer. So I'll click on that. And then this is the detailed information. We have a citation. So we've got a Cancer Letters paper that was published in 2017. So just sort of going back, if you click on the same one, it's record number 10 in the list. An FFPE-based prognostic signature to predict metastasis. If I'm going too fast or you want me to go over something, please let me know and I'll repeat it. Do you want to move forward? I can zoom, if that's helping. Yeah, so I forget to zoom as I move on the page because it reverts back to the one. So you can see there's a PubMed ID here for that paper. This has been produced by Steve Rosen in Duke, NUS, so it's National University of Singapore. And what you see we've got here is the 3 platforms. So these are effectively what's called the array design files. These are the details of those, and I can get, by clicking on here, all the gene names down. So I can pull down gene names and based upon their Affymetrix IDs, and I can get the sequence and all sorts of things in there. But I'm not going to focus on that just now. You can see we've got individual samples, and GEOD uses what's called a GSM reference. So GSM is the reference for every individual sample in that repository. So you've got GSE, which is the collection reference. You've got GSM, which is the individual reference. And you've got GPL, which is the platform reference. So theoretically, if you put those into Google, you can actually find that individual sample or those data sets, or that array. Now, there are some tools here. I can analyze with GO2R. These are like inbuilt platforms. We can start to do some analysis, but I want to do some of our own analysis because, like any mechanic or engineer, I want to get under the bonnet and start working with this. So if you see here, we've got the series matrix file. You can download that. If you click on it, that will download that file as a zip file. That will take you to this screen here. And that's the GPL 570, the one you want. That's the Affymetrix array. And you will be able to, if you double click on that, it's a GZ file, so you should be able to unzip that. You will need an unzip facility on your laptop, but 7-zip or WinZip are all free to download. Most archives zip the data up to save space and cut down the download times. Yeah. Yep, sure. What I'm doing is I go to the series matrix files. I didn't want to click on it directly because if everybody's clicking on it, it all causes a bit of a lag. So it's this one, the GPL 570, but if I wanted to look at other techniques, I've got those there. But this is the bigger data set anyway. So if you click on that, it should download. And what I've got in my downloads is I've got that gzip file. I'm sorry, I can't really, can I zoom on that? Well, yeah, a little bit. So I've got that gzip file, I unzip that, and I get my text file. Okay? So I've downloaded the data set. Now the advantage of working on Geo over some other platforms is they coordinate and curate all the data for you. Has everybody managed to download it? Are you able to see the file? Yeah, all the entries for the for Geode have a series matrix file. Yeah. So you can search more or less any system. You can find a series matrix file, and as long as it's the kind of array you want, you can download data and start to do some analysis on it. So I want to open that. So I'm going to just open it here. Oh, I have done already, but let's close that. I'm just going to open it in Excel because it's quite a visual way of looking at it. And I'm going to show you some analysis tools you can use in Excel. So if I click File, Open, I go to my unzipped matrix file and click Open. Now, because it's a tab delimited text file, I have to help it import. So we click on Delimited, and Next. We click on Tab, and then Next, and click on Finish. Yeah. So
the importing part.
Yep, sure. So if I click File, Open, yep, I want to click on the unzipped file. And because it's a tab delimited text file, it means that every cell is separated by a tab in the text file. You can also get comma delimited text files, and in fact you can get any kind of delimiters. I've even seen sort of semicolon delimiters and star delimiters. So if I click on that and click Open, you see the default is delimited. It's got here file origin Macintosh, but that's just because I opened it on a Macintosh. But you know, if you're working on a Windows machine, it'll just default to Windows. And then if I click Next, it automatically is detected as tab. But I could see, I could select comma, semicolon, space, whatever. And then I just click Next and Finish. And it will take a little while to load because it's 45 meg, but it shouldn't be too massive. So, has everybody managed to open the file? You okay there? Oh, okay. Has anybody else got an internet problem?
I think everyone is using the internet.
Sorry? Yeah, I think what's happening is you see everybody in this room is going through the same server, going to the US to the same server. So we haven't got the bandwidth for everybody to do that at the same time. So it will take a little bit of time. That's why I kind of downloaded it earlier, just to.
It's like loading, like it's taking some time. File is big, na, so.
Right, yeah. Yeah, I didn't have an FTP issue, but it might be that the server's kind of pinging back a response because it's a bit slow. So once this file's open, it's got a header. So you can see here, series title. So that's the title of the record. You can see all the things like the date of production, the date last updated, where it's come from, and so on. So we don't really need that, so we can delete those entire rows. And so if we just select those rows by clicking on the far left-hand side and right-click, delete, we now have moved those up. Now, if I click here in the top left-hand corner, that selects every cell in the array. And what the first thing I want to do is just double-click on the space between the rows, between the columns, to change the width so I can see what's going on in those data sets. So now you can see, we've got the records for these individuals. So the first one is an early stage colorectal cancer, female, aged 86, with negative metastasis. So in this study, it's positive versus negative metastasis, so we're interested in that row. So I'm just going to color that red. So what I'm going to do is I'm going to delete the headers that we don't need. So if I select, again, on the far left, that selects the entire row, and I click delete. I want the GSM reference, because that tells me which patient it is. And then I drag down. For now, I just delete everything right the way down to ID ref. Sure, I can delete that one as well.
Can you share the text file?
Yeah, do you have a USB stick? Yeah. What I'm going to do.
Save, you need to go to a text and then save link as.
No, like I don't save them.
Yeah, yeah. Right, so what I'll do is I'll open that file. Actually, I'll just take it onto the, so, just open this one here. That's your USB stick. And I'll just drag that into there.
Which file? Text file 100 MB.
Is it, might not be liking the formatting of it? Because it's a Mac?
Yes, it's your Mac.
Yeah. Just try and copy.
It's not sufficient to have a normal resistance.
No.
I see.
If somebody else has a USB stick, I could try different.
But still, জ্ঞানের ব্যাপারটা তো আছে না, এটি slow.
Yeah, so you just double click, and then if you just double click on it, it will take you to the FTP site.
So that is the reason it's not opening, because the FTP access is not given for the
Yeah, it should be. I mean, I just linked to it, but I'm not sure why it
You are
worked for some.
using the server of your PC, right?
I'm using Eduroam.
Yeah, that. Okay, you don't use it. That is the reason it's here.
No, I'm using IRTB wireless. At the minute.
That is what-
Yeah, that's strange. Yeah. Let's just try. No, I'll see if I've got the data drive. Has anybody else with a PC managed to download the file?
Yeah.
You have. I think you've got a Mac. Open the unzipped file, and it will go to delimited, and we want to select tab delimited and then finish. So you should see something a little bit like this once it's opened. Has everybody got there? Yeah. So the next thing to do is to select the row by moving the cursor over the cell to the left-hand side. You see how I'm doing that? And then if you click, hold down and drag, that selects the entire row. Because that's a header, I want to delete that. So I right click and delete. No. So open it from Excel. Has everybody got to that stage? So now what I want to do is select everything. So you see if I move the cursor over this top left corner here and click on it, it selects every row and every column. And what I want to do as well is just, because you see I can't see what's in the cells. So if I move the cursor between the columns, you see how it goes from a down arrow to a line with arrows either side. It might not be that clear, but if I double click on that, it stretches the width of the longest cell. So we can see the content of the cell. And then if I select from total RNA down to GSM 2179087, right click, delete. So now I just have my expression array data and the status, whether it was metastasis or not. So I'm just going to quickly reprofile those to take up less space in the spreadsheet. So if I, again, select all by clicking here, double click on the space between things. Now, the next thing I want to do is to sort so that I can bring all the metastasis negatives together and all the metastasis positives together. So to do that, if I click on cell B1, I want to select just the data from B1 down to the bottom right of the array. So to do that, if I select B1, I hold down Control, I hold down Shift and keep them held down, and then press down. That takes me to the bottom of the array. And then I keep them held down still and press the right arrow. That has now selected the whole array, but ignoring the blanks. So I'll do that again. So, just to repeat that, I select cell B1, I hold down Control and Shift, not letting go, and press down. That selects all the cells down. Keep it held down and press right. So that selects all the data, apart from column A. Now what I want to do is then I'm going to do a sort. So if I click Sort, Custom Sort, and what I want to do, instead of sorting by a column, I want to sort by the rows. So if I click Options, it'll be in a different place on a PC, but I want to sort, Options sort left to right. Okay. And then I want to sort them by row 1. So I've selected that range, I want to sort by row 1, I just click OK. And what it does is it brings all the negatives together and then all the positives together. So, but because I've selected B1 to the bottom right, not row A, it's left row A where it is, which is what I want to do. Okay, everybody managing to do that? So to do it, you click Sort, Custom Sort, Options, Sort from left to right, Okay, and then select the row you want to sort by. Has everybody managed to do that?
I can sort. So my past I'm using like you have said. So in here, how do you sort?
Yeah, so I'm not 100% sure of this. It might be the data or it might be edit sort. Sort.
Okay, this sir.
Yeah, but that's sorting.
So this is like
yeah. Sort left to right. Just out of interest, who's using Excel? Who's using Microsoft Excel? This is really Excel based, to be honest. So the next step is a little bit more complicated, but if you follow what I'm doing, you should be able to get it started. What we want to do is in Excel, there's a statistical analysis toolpack. So if we go to here, File, Properties. This is on a Mac. No, that's not it. How do we do it on a Mac? Yeah, so on a Mac, it's, if you go to the top, Tools, Excel add-ins. And there's something there called Analysis Toolpack. On a PC, it's, you go to the Excel Options window, and one question is yours, and then I can. So on a PC, if you go to File, Options, so you go to File, Options, Add-ins. And then at the bottom it says Manage Excel Add-ins. You click Go and click Analysis. And then this will appear under Data. Click Data. So did you catch that? On Windows, it's Options, File, Options, Add-ins. Manage add-ins, Excel add-ins, Go, Analysis tool. Let's start.
Could you please plug it? Can you get rid of that? Yeah.
No, I do. So, is everybody getting somewhere towards that direction? So, File, Options. So, it's the Windows here. It's the Windows button, and then you go to Excel Options right at the bottom,
Okay.
and then you go to Add-ins. And then Excel add-ins Go. And Analysis Toolpack. Okay? Right here? Okay. Okay, that's fine. You getting on okay? No, just the Analysis Toolpack. Yeah. Okay? So what this does, and I'm not sure whether I don't select it anyway, but it opens up an analysis toolpack, and what it will do is it'll appear if we go to data, so we go to data here, then you see data analysis comes up. Now again, it'll appear a bit differently on Windows, but you can see in here, you've got a whole range of tests from ANOVA, correlation, F-tests, histograms, random number generation, regression, which is the one we want, t-tests and z-tests. So if I click on regression, click OK. Now, what this has brought up is a dialog. So I'll cancel that and show you. So if you, under data, there is a data analysis tab. Everybody found that? Okay. So, you want to sort by this row? Yeah. So what I said is if you click here, you do Control Shift down, and I'll hold them down when you do it, yeah? And then while they're still held down, press the right arrow. So that's just selected all of the data, yeah. And then what we do here is sort, and we want this case, okay. And we want to sort by row 1. Row 10. So you see negative, 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 negative. Okay?
So I have managed to take all the metastasis into one row.
Okay, that's good.
But then how to take the negative?
So what you need to do is when you do if you do a regression in past, it's a stats analysis.
Yeah.
You need to select just one. You want a metastasis on the y-axis and a non-metastasis on the x-axis. So you find a pair of those.
Okay.
And then you do a regression,
Yeah.
and do what you want to include residuals and standard residuals in the output.
Okay.
I should be done in about 10, 15 minutes. So I just wanted to show them the print. Okay, so now we should have our data sorted. What we want to do, and we've got our analysis toolpack open, we want to do a regression. And that regression will put our, let's say we put the metastasis on the y-axis and the put the metastasis on the y-axis and put the non-metastasis on the x-axis. So every change will be with respect to metastasis. So if something's overexpressed, it'll be overexpression in metastasis. If it's underexpressed, it'll be underexpression in metastasis. So to do that, I want to go to data analysis, regression, click OK. And So our Y range is here, so I'll zip along here to the first metastasis positive. Keep going. It's a lot of replicates here, so. Right, so our first metastasis positive is in column CX. If I click the top cell of that and then do Control-Shift down, it selects all the cells in that column. And that's what's going to go on our Y-axis.
Control hit down. Yeah, okay.
Is everybody getting on okay with that? Yep. With what, sorry? Astrea. With R, you could do it with R, but that would involve people doing coding, so most people don't do coding. So you'd need to have a different workshop and training in R to do that. So what I've done there then is I've selected on the Y-axis these metastases. And we want to select a non-metastasis on the X-axis. So I clicked on the X range, click and do Control-Shift down. So I now have a plot of expression for metastasis against the expression for non-metastasis. And from this I want to select, I'll call it M1VN1. So metastasis 1 versus non-metastasis 1. And I want the residuals, standardized residuals, and the line fit plot. I'll try. It's let me do it. If you want to come closer to have a look, then please do. So just to recap, we're putting the metastasis on the Y-axis, the range of the cells, the non-metastasis on the X-axis, and then we're putting the results into a new worksheet called M1VN1. We're selecting residuals, standard residuals, and a line fit plot. So that's going to do a regression where we regress the first metastasis case against the first non-metastasis case. Okay. Is everybody getting on okay with that? So once we have that, we click OK, and it'll do a metastasis for us. Sorry, it'll do a regression for us. What we should get, once it's thought about it, is we get a new sheet. We have a line fit plot. So these are all our genes, and you can see some of our genes are upregulated with respect to metastasis. Some are downregulated with respect to metastasis. And from this, we have a standard residual value. So that value is how many standard deviations that gene is, or that point is, above or below the line. So if I just select all of those, I've got a minimum of 15. So that minimum of 15 means that there is one gene there that has 50 is underexpressed with respect to metastasis by 15 standard deviations. And if we look at the max is 10.23, so there's one gene that's 10 standard deviations above the line. So you see, we're seeing now which genes are up and down regulated and by how much. So what I want to do now is, if I do Control C, I want to find out what they are. Obviously, I could do there's a shorter time. I could regress every single one of these against every single other one. It would take me a long time, but I'd have a pretty good overview of what's happening in that population. But for now, I just want to quickly have a look at those residuals. I'm going to paste them next to my data. So what I'm going to do now is, if I sort based upon that residual, I can see which genes are overexpressed and which genes are underexpressed. And because I've got the Affymetrix code for those, I can find out what they are. So, sorry, it's still sorting on rows. I need to check it from top to bottom this time, and I want to sort it based upon column EW, smallest to largest. So it'll take a little time, it's got a lot to sort, but it will give me the top overexpressed genes in metastasis and the top underexpressed genes in metastasis. So, can everybody see what I've done so far there? You've done a regression, we've got the standard residuals. The standard residuals tell us how over or underexpressed a gene is. And from that, we can explore what those genes are doing. My sort doesn't seem to have worked. The whole thing is fitted to equation, but basically when we do a regression of row 1 versus row 40, that will give us an equation, and the fit to the equation gives us the residual, and that residual tells us whether a gene is over or underexpressed. We don't define the equation, that's why we're doing regression. The regression defines the equation. Right, so I've sorted that, and I can see in this quick experiment these are our negative genes, and you can see these go from about 5 or 6 standard deviations down the line. If we look at the distribution of those, I'll do a plot. You can see we've got these hooks at either end. So these are the down-regulated genes, and there's a small number of highly down-regulated genes. And then we've got the up-regulated genes, and there's a small number of up-regulated genes. So what are we interested in? Well, sorry, I'm going to zip across there. Our top genes are these. So I'm just going to take 10 of those. So these are the ones that are downregulated in metastasis in colorectal cancer, and I'll copy those. Now what I want to do is find out what they are. So to do that, I go to a web page called Gene and this is part of Gene Cards. What's going on there? That's not what I expected. Gene anot home. Yeah, it's a gene anot search, no? Strange. Just Google it instead. So yeah, you want gene anot home. If you Google it, it comes up as a top hit, but if you use Yahoo, it doesn't. Something to do with browsers there. So if I zoom in on that, you can see what we've got. So we have effectively a way of taking our probe names into this, and it'll identify what they are as genes. So I'll paste those and click Submit Query. Now, there's a bug in this page, so what immediately happens is it comes up with the interface again. I'll submit them again. Now, this goes to gene cards, takes the annotation name and identifies the gene name for that. So you can see here we're getting these are the down-regulated genes. We can see we've got olf M1, so alpha-medin, exist, 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 exist. So there's lots of repeats of that. Reg1b, H19, and a repeat of that, and then some D5. So if we want to know about one of these, if we click on the hotspot or the link for that, you see, and you want H133 plus 2. Yeah. So you can see here I've pulled up this SAMD5 gene. I've got all the information associated with it, where it's located. So it's on Q24.3 on chromosome 6. And you can see I can get a lot of information out from this particular gene. So what I also want to do is I want to know what I'm going to do is I want to know what these genes do, and I want to understand a little bit more about their ontology. So if I copy this table, I can put it into Excel. I paste as special as values. Unicode text, that'll do it. And I can get the gene list based upon the ID from there. So what I'm going to do is I'm going to copy that and paste that and sort it. So if I want to know more about those genes, I can put them into an ontological database. So one of the ways to do that is to go to something like Panther ontologies. So if I again go to Chrome, click a new tab. And I want to search now for Panther on gene ontology. And I'm going to select humans and search for good Panther tools, multiple gene list. So now if I click all, it brings up all the genes And I can see things like what are the, click on this histogram here. Look at the molecular function of those. So I'll just maximize that, zoom in. So you can see here, of these genes, there's only a small number. We've got binding and molecular translation activities. We can also search for biological processes. And that gives us regulation, population proliferation, cellular processes, multi-organism processes in response to stimuli. We can look at the cellular components, so where are they found in the cell. So these are extracellular predominantly, completely in fact. And we can look at the pathway that they fall in. So there's no hits in the pathway, a low number of things. So that is one way of finding out what these genes do. We can also go to something called string. And some people have shown results for this. So we can drop the short name for the gene into here and click search. We want to check Homo sapiens, click continue. Select these, click continue. And you can see here we've got relatively few hits. But there's a potential interaction between OLFM1 and Reg1b. If I want to find out where that is, I just click on the link between them and it tells me details of that and it gives me a score and so I can get information on that. One of the things we can do with string, though, is we can grow the network. So we can add layers in to see what's interacting with the things, and if those are boosting interactions. So if I click more, now you see it's added in a number of extra proteins. I can click more again. And the idea is really, if we keep clicking more, do we get to a point where we've included everything? What seems to be happening here is we've got a very strongly regulated motif, but these things are still independent. But this is basically a protein interaction based upon the literature, based upon hypothetical binding, based upon energetics of proteins as well. So from this, we can see whether things are interacting as well. So something else we can look at, and I'm going to be relatively quick now because I don't want to keep you from your lunch, is something called GeneGo. No, Gene Mania, sorry. So Google Gene Mania. I don't know who decides the names for these things, but so what I can do is brings me to a splash page. You can see up here it's selected the species as human. If I right click on that and click paste, it's going to add my genes in. And I click search. So what this is doing is looking at the interaction at the gene or transcript level rather than the protein level. And you can see now I've got various links. I can highlight those links by moving over a particular thing. I can zoom in, zoom out of that. And if I click on a link, I can get more information. So it takes me to the paper which has found that interaction. If I click on there, so there's various things I can click on. It gives me the strength of interaction, so that bar along the bottom tells me how strong it is. And by clicking on the link, it takes me to the paper. Now, you can do, this is all just sort of high level or low level activity, but once you've got this, you can start to add filters in, you can start to export. Now, these are tools that the best way for you to learn is to have a play with, really. So those three things, string, gene mania, and Panther, give you quite a good overview. You could also go to something like David, which is also an ontological search. But also, don't be afraid of typing the gene name and the thing you're interested in into PubMed and see what you get. Okay, does anybody have any questions?
How can we compare two different fields? Like if we have the data, if they are overlap or not, do we do them separately and manually put them?
The only way you can compare two separate species is to look at homology between them and just compare the homologous features. So say you were comparing mouse and man, you would need to look at the genes or transcripts where there's a high level of homology. But then you'd also have to co-normalize the cell files as well, because you would probably have a mouse data set and a man data set. You need to normalize those together from the cell files and then strip out anything where there is not good homology. Otherwise, you'll just end up finding a set of things for mouse and a set of things for man. So you need to transform based upon homology. If you look, there was a paper which we produced a relatively long time ago in sorry, I can't remember. If you Google me and rat-human homology, then there's a paper we produced where we worked with a company to look at the homology between rat and human, and just then started to analyze the homologous activity. Sorry. +1. Yeah. So that would be the strategy. Any other questions?
Just make an observation that one of the genes was access.
Mm.
I think that's the one that affects the expressor.
Okay.
So I guess the notation that file from the design as well.
Yeah. Obviously, we just looked at the top 10 and then did one comparison. So that could have actually been, in that state, we compared a male with a female. Yeah, exactly. Yeah. Okay. Right, so thank you very much.